Welcome to Neurosalience, the OHBM podcast.
Welcome to the Organization for Human Brain Mapping Neurosalience podcast. I'm Peter Bandettini, and here I explore brain imaging and modeling with neuroscientists from all over the world. Dr. Michael Milham was one of the panelists at the OHBM meeting last June in Seoul, and today he's here to talk more on the many challenges and opportunities in big data MRI and fMRI, and on the characterization of individual differences, topics that Michael has been thinking and working intensely on for decades. Dr. Milham is the Chief Science Officer and the founding director of the Center for the Developing Brain at the Child Mind Institute and director of the Center for Biomedical Imaging and Neuromodulation at the Nathan Kline Institute for Psychiatric Research. He also fits in his practice as a child and adolescent psychiatrist. Michael's research interests include investigating how functional and structural connectivity in the brain may underlie differing forms of mental illness in children and adults. He's recently embraced the challenge of collecting rich multimodal behavioral data for more precise delineation of pathological phenotypes. After receiving his undergraduate degree from Duke University and his MD and PhD degrees from the University of Illinois at Urbana-Champaign and the Beckman Institute, he moved to NYU to work with Javier Castellanos during his residency. During this time, he helped establish functional neuroimaging at NYU and Nathan Kline. He quickly understood that to move fMRI towards clinical relevancy and to advance the field in general, large multi-center databases were required. He collaborated with Bharat Biswal in establishing the 1,000 connectome dataset in 2010, highlighted in the landmark PNAS paper Towards Discovery Science in Human Brain Function. Since then, he has gone on to lead the establishment of many consortia and big data sets, including the Autism Brain Imaging Exchange, otherwise known as ABIDE, the ADHD 200 dataset, the Rockland sample, the Consortium for Reliability and Reproducibility, and the Child Mind Institute Healthy Brain Network, which includes 10,000 participants. Michael's one of the most highly cited brain imagers in recent years and was recipient of the OHBM Early Career Investigator Award in 2014. Here we discuss his unique career path, his many collaborators, the goals and challenges of big data collection and analysis, the importance of scanning each individual for a sufficient amount of time, and the growing trend in deep phenotyping to leverage more information from the fMRI studies. Michael's passion, insight, and intensity come through in this amazing conversation. I hope you enjoy it. Welcome to the OHBM Neurosalience podcast. Today I have Michael Milham. It's an honor to have, taking a little bit of time out of his probably incredibly busy schedule to come on and talk about everything he's been doing, from his early career to his databases to his cutting edge work on really sort of characterizing variability in the data and stability and replicability and that type of thing. And so looking forward to the conversation. Thanks for joining.
No. Thanks for having me.
All right. So just to begin, I always like to start just to give a little bit of a tiny origin story of all the people who come on. And I've known you for years and years, and it seems that I feel like you emerged when I became aware of you. You were already pretty senior and already sort of put in charge of at Nathan Kline and NYU and in sort of leadership positions already. And so how did this all begin? So how even going back to when you first became interested in either medicine or brain imaging and going from there.
My first interests probably were more in the clinical direction. And if you asked me in high school, my initial thoughts were I'd wanted to be a doctor. And that's where my mindset was going. In my junior year, there was an after school program that I became aware of where you could go and do research, it got you out of class a couple periods earlier, which had appealed to me. And I figured it'll look good for college applications. I didn't really have any idea really where I was thinking to go to college or anything, but it seemed reasonable enough. So I said, why not try? And what wound up happening is it started off really boring. I was sitting in a room doing nothing, watching some people hand code things on video, and then one of the two folks whose lab I was in came by and spoke to me a little bit and realized that I had some programming experience from when I was younger. And so they quickly had me start learning more with Quick Basic and working on doing basically behavioral viewing tasks, trying to understand a visual preference because there was a lab focus on infant development and infant brain development. And
Yeah.
then from there, they brought up the issue of a Westinghouse project, which I guess now is
Wow.
called Regeneron. I think prior to that, Intel and so forth,
Yeah.
and basically said I would need to learn to code in C in order to do this, and had me thinking about how to use EEG, steady state evoked potentials, to track an infant's ability to detect a structure from motion
Hmm.
in an objective fashion.
Yeah.
So that got me to really delve in and start to learn a lot more, obviously, about electrophysiology, a lot more about coding, but also about the brain, and become more interested in the question of how do you measure something objectively, which is otherwise difficult and subject to either overly laborsome approaches or just purely subjective characterizations.
Yeah.
And so that experience really became a game changer for me. I wound up working both summers. Initially, it was a volunteer program. They were like, look, we don't want you going and working. We want you to work here. And they had me work throughout the year as well. And then actually all my college summers, I would go back there. And it really became a rich training experience and learning experience for me. So then when I went to college, I initially was going to stick in the clinical path, but my mentor from high school told me if someone tells you they want you to do database work, say you don't do that. You have too many skills. And
Yeah.
so
Yeah.
I happened to there was investigator John March at Duke where I went to undergrad, who he had posted something that sounded kind of exciting. I called him up and he explained to me a lot of it was about data entry and so forth. And I said, I don't do that. And so next thing that happened is I wound up, he told me, come on in and speak. And I spent college, he'd wanted to really think about how do you bring cognitive neuroscience to the study of mental health. And he's particularly focused on child adolescent mental health. And he would weekly mentor me on issues in child adolescent mental health. And at the same time started pointing out functional MRI to me, and I was also working with EEG there. And but really designing initially just first implementing with a grad student at the time, and then taking all the tasks in a battery, and then running participants, and then really wound up doing the design of all the analyses, so that in that process, there was a bit of a conversion of thought, where it's the first time I really thought of research as a career path.
Yeah.
I also did a second major in computer sciences because my high school mentor was always on me about you need to pick up those skills, not to go become a programmer because it was the web boom, but in order to become a really good researcher, you have to do this.
That's
So that really prepared me, and then from there, I eventually got talked into MD PhD. So I went to University of Illinois, where I did MD PhD in the lab of Marie Banich. That was very focused on attentional control, thinking through the anterior cingulate, dorsal prefrontal, relative contributions. But it was really an invaluable experience because I'm helping to build a imaging program up at the Beckman Institute from scratch because there wasn't an imaging program.
wow. Okay.
And so that really gave me a chance to dive into everything with my own hands, and it was an invaluable experience. And I got to I ran way too many Stroop experiments for my own personal sanity. But it also really gave me access to the scanner in a volume that I could really delve into questions and not be afraid about if a question is wrong or not.
Yeah.
We were really able to explore a bit more and not always be right, which was great. And Maria was a wonderful mentor and helped me think through much more of how to put things together and questions to choose, and a lot of balance of having to write all the software to do the analyses or visualize or explore versus so folks into cognitive neuroscience. So, from there, the biggest challenge was I was still a little disillusioned about where is this going. But then I went and did a month in my last year of medical school over at National Institute of Mental Health in the lab of Danny Pine, where,
Oh, I didn't know that. Yeah.
oh yeah,
Yeah.
no, Danny's things was what do you think you could do in a month? If you want to do analysis. And I was like, well, so one of my colleagues at the Beckman, Stan Colcomb, was so obsessed with voxel-based morphometry at the time. And
Yeah.
I was like, well, he's doing that in aging. I was like, why don't you give me all of your pediatric data, and let's I'll put a pipeline together, and let's see what we can do, like anxiety disorders and so forth in kids. And the first thing I learned was how much head motion was in that data. So all of a sudden, you couldn't just sit there and take SPM and just start applying. I had to sit there, start really thinking through how to deal with the motion, doing way more time with QC. A lot of time was just being like, wow, this is, and really learn to the realities of that. But it wound up being incredibly productive. We wound up getting a few papers out of that work, both looking at pediatric anxiety disorders and pediatric bipolar. And that really gave me much more appreciation of why my mentors got me to do all that. And
Okay.
then came for where to go to residency and fellowship. Everyone told me Javier Castellanos is the person in New York that I need to meet, and that the two of us would be great. Of course, Javier's first few emails did not respond other than yeah, it sounds great. And then there was a fellow, Dan Dickstein, who was at NIMH, who was like, wait, I know Javier. He called him and said, you really actually need to look. And next thing you know, Javier, I get all these emails, and everyone's now talking. And Javier's like, look, I want you to come to NYU, and I don't want you sitting in emergency rooms moonlighting. I want you to be building an imaging group. And
That's
so, when I went to NYU, what wound up happening is Javier and I worked it out where I was, in residency, but my second job was to be his chief of functional imaging, which
yeah.
I was like, you don't have a functional imaging group in your institute. He's like, no, we don't, but that's why you're here. So I wound up throughout residency and fellowship, basically building the functional imaging group. He was able to sit there and get me a couple research assistants, and then we recruiting postdocs. So the first one being Claire Kelly, and then there was Lucina Udine, and then Mar Jimenez and Sinyan Zuo. And so over the course of residency and fellowship, I was also working to, I was already keeping very active in building the resting state up front for that, which resting state was so much serendipitous as to how we wound up taking that direction.
Yeah.
Initially, I was obsessed with having the more comprehensive understanding of working memory
Okay.
and ADHD, and also reward and circuitry and salience and so forth. But Javier kept pointing at Brock Biswell's work
Yep.
and talking about the fluctuations, because Javier was at the time obsessed with fluctuations in behavior. And he
Yeah.
had noticed that the fluctuations in behavior that you see in ADHD, the frequency matched that of the resting state signals variations.
Interesting.
And so
Okay.
he'd always talk to me about this. And he
Yeah.
was looking at using fNIRS and so forth for this with Adriana DiMartino. And I kind of kept scratching my head and be like, oh, I'm not sure about that. And then, Daniel Margulies had entered the picture in his own somewhat unique way.
Yeah.
Uh,
As he does. Yeah.
yeah, exactly. And, first we were looking at variations in the frequency of tics in Tourette's and so forth. And I was not really bought in on the whole fluctuation story at the time yet. And then Daniel one day popped up with Mike Fox's paper, as well as Francis' paper, the 2005 papers looking at default network. And at that time, we were doing ALE meta-analyses with ADHD and putting some of those out and building different fronts. But
Yeah.
I was happened to be at the Nathan Klein as a resident doing a rotation and had some time on my hands. So I just quickly took a bunch of task data that we were collecting and analyzed everything as I would resting state.
Yeah.
And I was blown away by how large the effects were.
Yeah.
And so that quickly, then we immediately started just drilling at outstanding questions in my mind. Like, so their anterior cingulate cortex. And with Maria, I would spend so much time trying to delineate conflict monitoring versus response selection, all these things, and sub-regions within cingulate, because it was driving me crazy in the literature that everyone treated cingulate like just cingulate. And
Yeah.
so that's where we did the systematic seeding paper with Daniel, which was one of our first, really with the idea of neurosurgery where you lay a grid down. And so we said, let's do that in cingulate. And that became very exciting to see that in a small sample, we could replicate so much of what you get out of meta-analyses in the literature. You capture what you could see in circuitry at a level you could think of in non-humans,
Yeah.
primates and so forth. But there, so that got us excited, and we started getting this path. Then it was like, oh, let's do striatum. So that's where with Adriana DiMartino, we had already been working on some other work looking at ventral striatum and ADHD. So we went in, striatum, and then with Amy Roy, we wound up doing looking at amygdala, and then Margulies was like seeing her all night long, like trying to just seed everything possible. Whereas you have like
Yeah.
a Claire Kelly, who is much more like, let's think about networks and cognitive science. And Lucina
Yeah.
was also more in that domain as well. But the precuneus started emerging, of course, everyone's site, and that became something because the default network
Yeah.
Javier was very focused on with ADHD and the default network hypothesis. He's proving ADHD, so
Yeah.
we suddenly jumped into looking at the default network and its interactions in ADHD, as well as then saying precuneus and looking at not only can we look at this in humans, but because we were so inspired by the work of Mike Petrides in the non-human world, and we actually Justin Vincent happened to be visiting us at the time, and we hung out with Justin. He's like, if you
Oh,
want
yeah.
non-human data, the data I just published, you can get access to that. And so I was like, I called Daniel. I was like, you have to speak to Justin, and they wound up with eventually becoming that translational paper. But this was all like a very
Wow.
for us, it was very much like constantly testing ideas.
Yep.
And then we stepped into the whole question of individual differences, which, there we had two driving forces. One is we had Don Klein at the time, who would sit there, and every time Javier and I would send him an Ames page for a grant, Don would say, anything that is this robust is either an artifact or biologically unmeaningful unless you prove
Yeah.
this otherwise. So one day, Javier and I were sitting, I was like, how do we just put this to rest? And he's like, we would have to do a test retest. I was like, great, we're going to do test retest. So we went in and did really one of the first test retest studies in resting state of fMRI, the Shahzad study.
Yeah.
And,
What year was that? Was that what year was that?
It's like 2008, maybe?
2008. Yeah. Okay.
Yeah, 2008 or 9, I think 8 is in cerebral cortex. And it was interesting because there's so many ways one can go at the time. We focused more heavily on the regions we were most obsessed with at the moment, being frontal parietal cortex and default network, which is interesting because those wind up being the more reliable ones in the brain. So we felt a little falsely reassured by the areas we were looking
Yeah.
at.
Yeah. Yeah.
And I mean, there were. It's funny, that paper. There were so many so many tables and figures in that paper, and there was a number that people got me to like not include because they're like, you're getting crazy with this.
Yeah.
And there was a lot of internal back and forth, but like there's just so much. That was just, we didn't realize how big that set of questions would be really for the next decade.
Yeah.
But it also coincided with us thinking about individual differences because like one day. Actually, Zarar and I had a conversation. He was talking about non-human, actually, it was rodents and self-licking behaviors and how people measure things. And we're like, well, why not go and see if we can relate things to individual differences in resting state fMRI? And so we called a bunch of subjects back, and I told everyone to go after get the questionnaires you care about. Let's characterize these folks. on the multiple questionnaires. And so we went up with like Adriana DiMartino had the SRS, looking at autistic traits, which became an American Journal paper. We had Jonathan Adelstein, who did the Neo5, which became a big thing in the group because everyone started doing their own Neos. And I told them, well, like, if you're going to take this test yourself, let's not share it. Next thing you know, I walk in and they have distributions of all their scores put up, and it was an interesting group. But that's where we then started looking, and so then we started saying this is reliable. This is it seems to be sensitive to picking up differing effects, and the results kept being sensible. A lot of it was because of the background, understanding of cognitive neuroscience. You could go and ask the questions in more targeted fashions. And so that really became, we were excited, but then the question was like, what next? Which was probably towards the end of my fellowship. By the time I finished fellowship in 2009, was when I finished that the medical training. So in 2008 timeline, we had at that point really delved in, and we had started getting to this question of, well, what does it look like across scanners or across studies? Like, how do we show that you could replicate? Because we were struck by, we kept writing like how replicable patterns are across studies. One day we were sitting there asking that question, and we realized that the right person to get involved with that would be Bharat Biswal. So I called Bharat and said, Bharat, you have access to lots of data and different people. I have some friends. Like, why don't we maybe see if we could get us all to work together to just look at, do we all get the same patterns? And of course, Bharat said, yeah, that sounds great. It's on the list of things that they always want to do. So we got it. We had started it early in 2008, actually. We got maybe like 5, 6 data sets, 7 data sets. And then at the first resting state workshop, which was in Magdeburg, Rob presented the data. And that's when in the room, what I realized was the 2 questions, which was, do you want our data? And also, can we access that data? And so that's where Brad and I sat down. We're like, okay, this would be great to build this huge thing. And obviously, we don't want to be the owners of the world's largest data. So, we went through that process, and we had to take on the question of, can you aggregate the data meaningfully? And so that was really the heart of the message we were trying to drive through in the Biswal paper. And we went through the many kind of pains you go through in starting, data aggregation and pulling in all that data with really no infrastructure. That's where folks like Martin Menes and Zuo, like just an endless amount of effort went into figuring out how to actually do that. But that became really defining in my career for where I would go, like once done with fellowship, but really for the next decade and a half.
No, that paper actually was sort of a landmark paper of the first, like, okay, so we can pool our data and, and then, right, like you said, sort of addressing the issues of pooling the data and then trying to draw something of it and out of it. And that was completely unconstrained. That was different groups doing completely different, parameters, their pulse sequences, everything. But and that was published in 2010 in PNAS. Just one really quick question. So going back, so you're doing all this during your fellowship and or your residency. That's amazing in itself, and it was great that Javier gave you the freedom to do that. One just mention about Javier is that I remember meeting him early on, maybe at that Magdeburg meeting, and I just got a sense that he really he got it. He kind of knew what he was doing. He was asking the right questions. He really had a good kind of this visceral feel of fMRI that a lot of people didn't seem like they had at that time. So seems like he was ahead of the curve in that regard of his insight.
Javier is the guy who's always kind of looking where other people aren't thinking to look. So his obsession with the fluctuations in the MR signal, and he would have these meetings where he was pulling together people looking at fluctuations in temperature, fluctuations in neurotransmitter release, fluctuations in the pupil, which is funny because at the time I was like, Javier, can we just focus on one thing? and Javier and I were always a very good balance for each other because he tends to kind of be much more, let's just think outside the bounds to over there and over here. And what if we put this and that together? And then which is not my first natural inclination. Whereas I'm more of like, then I'd be, no, that's not how you do that. If we're going to do that, and then I'd be the one to be, here's what it would take if we actually want to do that, and why. And we'd kind of go back and forth. And it was really a wonderful partnership that way. And,
Yeah. Yeah.
there were, we were also blessed with the number of, with the talents that we wound up having come to that group back at NYU, in those days.
So what scanner? So you were at NYU, or you were at? So what? Like the scanner. It sounds like you had a ton of freedom in terms of using the scanner, and it sounds like you said, like there's, with any group that is takes off like this, it's like just the right combination of people, right combination of sort of, you know, free to sort of ask any questions you want, to try things, to iterate, and it sounds like you had that. So, were you at Nathan Klein, or yeah, where were you?
Well,
Uh,
yeah. So the way it worked was, I was at NYU as a resident fellow from 2004 to 2009, and then I was faculty there from 2009 to 11. NKI started working at in 2010,
okay.
and because and then in 2011, I would leave NYU to for the Child Mind Institute in order to build a research program there, while at the same time staying at the Nathan Klein Institute.
Yeah.
And so when I was at NYU, we would work on the Allegra that was downtown,
Okay.
and then which there, there folks tended to really like daytime hours for studies, whereas we were good with paying, with having lower rates and working after 5. So we did do well as far as data collection amounts and so forth. And what once we pivoted from the focus on a very cognitive task focus, test-based fMRI approaches to resting state, it also brought about shifts in our thinking a bit and what we were after. But it was also a little strange because so much what we were initially collecting, we never even wound up publishing. It was the smaller part of the sessions that we did, or otherwise then we repurposed things in different ways. So
Yeah.
some of those pivots were incredibly rewarding, but on the other hand, some things it's like, oh, okay, we just spent a bunch of time designing those studies. We did all that. I really love that task, and no one's ever going to see it
Yeah.
because
Yeah.
it became so productive, and there were so many things that we were trying. And the whole thing also was there's a big focus on trying to push the analytic innovation. And
Yeah.
when you start deciding that you're going to go after 1,000 data sets, that's you have to really take a step back and
Yeah.
every piece of it go through and spend all this time on registration and segmentation and trying to
Yep.
think why this didn't work or this did, and then all the kind of headaches you deal with in a period where it was nifty format only, and also
Yep.
where different packages, everyone
Yeah.
used
Oh,
nifty, but they all used it differently. And
yeah.
there were things like in that Biswell paper, like Martin Manders had to reanalyze everything
Oh man.
after the initial submission when we realized that one of the data sets didn't have the TR set right in their file, and compare, and then came also then having to analyze, because when we had shared the data with the community, I think it was Daniel Glenn at NIH, was
Yeah.
he's the one that noticed that one or two, that a couple of the sites when he was looking at asymmetries, that he's like, these sites, one site was completely flipped left right, and another site had half the data left right and the other one right left. And I was
Oh
like,
my
there's
God.
no way. And then it called up the person who cited it, wasn't like, that data was extracted by 2 different people. I was like, oh my God. And so that we then sat there and everything had to be redone. And you know that with such small amounts of a large data set, it's not going to matter, but you still have to do it.
You still have to do it. Right.
Yeah,
Right.
exactly. And so that turns into a lot of suffering for those involved.
Yeah.
And so it was a extremely hardworking group, which made it, the group also managed to have a lot of fun too in the process. But it was a very, when you're trying to do these things with larger data sets, or more complex designs, or even just thinking through if you want to go really wild with seeding and developing different methods, you didn't have anything turnkey to really help you do it.
Yeah.
You
Yeah.
wound up coding it all up yourself, which is great learning, but
Yeah.
it also it made it where it's very punishing when we had to redo things.
Right. Right. And then on top of that, there's nobody who actually thought, oh, I'm going to be setting a standard. And so, professional programmers sort of think like this, they sort of embrace it, but you're just trying to figure out something that, that will work. And yeah, that's a nice example of how in this case having standards really saves a lot of work and a lot of time. And
Yeah, and also your point as far as what you don't realize in a moment. So like some things, suddenly people are using some of the nomenclatures that we had set things up with, and we realize they're taking it very serious, and we're thinking we came up with that designation in the middle of the night just to.
right.
So it's it makes you more aware, and you start to realize, okay, wait, you have to really think things through, or
Yep.
be very careful to say, don't overtake serious this particular designation. Or even like, one of the things that people would comment to me about is your methods are changing so much from one paper to the next. And I'm like, because we're still figuring out how to actually handle all this resting state data. We're trying to figure out what's the best way to map these areas, or what's the best way to characterize. And
Yeah.
do we want to think about seat-based approaches? Do we want to be thinking more network level? Do we want to do individual level parcellation, group level parcellation?
Yep.
And we would have a lot of things that would be guiding our thinking, which wouldn't always come through. Like with seed-based, we were seeing all sorts of parallels of seed-based with the developing brain. Actually, because I was playing at that time, I became obsessed with for a while with structural covariance.
Yeah. Okay.
And when we were using seeds for structural covariance, you very quickly started to appreciate the amount of variation that could be within a seed region, and
Yeah.
how that variance could actually drive so much of what you're seeing outside.
Yes.
And
Yeah.
and you start then actually with some of the work I'd done with Cameron Craddock, we started realizing the seed differences could also be about the differences in connectivity you're seeing for seeds could be more about the fit of the
Yep.
seed ROI for the functional area you're thinking, than actually the difference, the connection. And then we sat there, and also with Xiaogan Yan, with Xiaogan, which was just a little later in about 2011, 12, when Xiaogan was at NKI and CMI.
Yeah.
We started like looking and comparing different pipelines. And the time we had started building the CPAC pipeline, and Xiaogan had DPARS, and I mean, it was always a thing that always drove me crazy. Is we'd always have so many pipelines among even different postdocs in our team would be using different pipelines.
Yeah. Oh, God.
And CPAC was my solution to try to make the pipeline that you could configure to everything, but still, we would still have different pipelines. It's probably
Yes.
to this day. But like we would start noticing that certain data sets, when we started looking, you would see correlations of 0.8, 0.8, for each of the subjects. And then all of a sudden, you would see there's a subject that's 0.4
Yep.
for the map that's produced.
Yeah.
And it would happen specifically for seed-based. And then we start looking at why. And then you would see, you put something into what you think is precunious and in standard space, and then there's a little variance in registration that's happening. And what's happening is you're actually now in cuneous in that subject. So because you're near a sulcus, you're sitting there and moving, or because you're near a functional boundary, and you'd be really looking at this and realizing,
Yep.
oh, this is built into people's data. This is messy, and
Yeah.
which was always hard to get anyone to work on that specific project, because that's like not overly appealing to folks.
Yeah.
To really, because but we used to do these style questions and really spend a lot of time.
I think that's an important, I think it's still an issue now. I think that,
Yes.
you know, parcellation is a solution to sort of analyzing, whole brain resting state data. But I always worry about exactly, along those lines of what you're saying with parcellation. If you have a standard template that you warp other brains into, or even if you do individual, it's messy, and you always wonder how much you're partial voluming and how much the signal's mixed, and how much is that driving the results. And it seems that there's it's still evolving the field in terms of what is the best solution? Is parcellation really the best thing to do, or is it, you know,
Yeah, no, that is a tricky one, and I've gone. I mean, over the years, I've definitely. I think there's different. I think it's what comes down to purpose. If you're trying to ask a group level question, you could probably do, and try to understand about connectivity and so forth. Like, you could maybe do fine. If you really want to start asking about individual differences and so forth, or go individual specific, you really need to be very careful about the question you're asking and what you're attributing to function as opposed to structure. And that's something that even many years ago, like Cameron Craddock spent a lot of time with the twin data set that we had and did some really beautiful work, but you could not get away from the function structure concerns.
yeah,
And you have to I mean, even when you think back to VBM, with VBM, what you're doing is taking imperfections in registration and using it to get meaningful insights.
yep,
So when you deal with with functional MRI, all you care about is the question of, does it work? Does it relate to something? You could
yeah,
forgive all that and say we don't fully know what drives it. Which, I mean, you're doing with the bolt signal, so
yeah,
it's
yeah,
all
yeah. Right.
very
There's
speculation.
layers of uncertainty.
Yeah, but otherwise, if you really want to understand you have to be very careful. And I think that's why we definitely have been much more sparing in our usage of C-based approaches. And if so, we usually first have tried to base it upon the more specific data set. And I think that just hits a number of different questions. And part of it is, why are we going to C-base? Or parcellation approaches. Like, I mean, like, for parcellation, like, why do we need to? And that's computational
Right.
expense
Yeah,
largely.
yeah,
So I mean, like, they're like in the grading from Karl Heinz Nenning and our team over at NKI. He recently published an approach that lets you do estimation of the gradients at a much higher resolution before going to parcellation.
yeah,
And we
yeah,
looked at the question of, if you're doing vertex-wise estimation versus doing parcellation, like if you parcel after estimation versus first parcellating and then doing estimation, you do take it is different. You do take a hit by parcellating first.
okay,
So
okay,
it's like any other data reduction. Sometimes the question is when, and
yep,
it could be that we're throwing information away by parcellating so quickly, which for us, that, so Ting Zhu's work, with the first paper she had done with us, was really looking at aerial organization, reliability of the aerial organization. And we were first starting at parcels, but then realizing, well, but wait, let's just look at the local organization and what more gradient of change there.
yeah,
And we started realizing that that's highly reliable. But if that's reliable, if your aerial organization is reliable, then that means if you're going to just apply parcels to it, you're inherently biasing the deck,
yeah,
because it's going to be, you can get effects that are all about the parcels fit to the individual,
yeah,
and
yeah,
you right back there. And so, and then we've played different games, like with bagging, with oculonicolitis, and we spent a lot of time going through those, but it's an imperfect answer at best. And
yeah.
interpretation, it really needs to be, people need to be careful with what they think it actually means.
Yeah, I mean, I remember, right, when Emily Finn came out with her brain fingerprinting paper. I always worried a little bit that that it was somehow driven by the structure, the connectivity profile, because there was a standard parcellation, and I've become more convinced that it was driven by the correlation, which still opens up other questions of how is the correlation driven. I mean, you could have a high correlation for the amplitude of one area is higher, and it might be, or it might be less noise, or other. That's another question. But
Mhm.
simply capturing certain structures better because of the registration with the underlying structure might somewhat bias the data. I mean, it's not like it's driven.
Absolutely.
Yeah,
And it's also tricky when people apply network masks, because we know that the networks vary. The function structure, it's not one to one.
yeah,
I remember many years ago, Randy Buckner expressed concerns about stepping into individual difference research in fMRI, basically saying, that the function and structure coupling is not there, especially once you go to higher order regions. So what are you actually measuring? And I think at the time I was first just happy to see relationships be like,
Yeah.
there's feasibility of this thing. Over time, I definitely became much more in tune with that level of concerns. And, part of the thing that for me really started to resonate was it could then goes down to the whole question of are we even getting enough data? And
Yeah.
that's where, when we started at CMI, we were building the Healthy Brain Network, and the whole idea of going after aspiration of 10,000 subjects, brain imaging, a whole range of modalities, EEG, eye tracking, and so forth. Children and adolescents with one or more mental health disorders, like 90% of them.
Yes.
And that was exciting to us. But the thing was, we were first just trying to figure out what imaging.
Yeah.
And what happened there is, like, Cameron Craddock and I, we were sitting there looking at this, and when we were trying to, people were who's using resting state, who's doing task, who's doing movie. Lucas Power got us very excited about EEG movie, naturalist viewing EEG, and we were doing some cool stuff with the movies there, with interceptor correlation and EEG. So we put together what was called the Healthy Brain Network Serial Scanning Initiative, and that was, so before we started, it's probably about like 2014 or 15. It would have been 15. We would have done this. We had gotten 14 individuals, if I remember correctly, who each did 12 scan sessions. And in each scan session, we had one rest, one, I believe it was Inscapes, one task, and one movie. We asked the question of, from reliability perspective, of how much does scan state matter?
Yeah.
And it was a humbling answer that we got, because it didn't matter a heck of a lot relative to just the variation you get between sessions. And so we quickly saw the biggest thing is just get as much data as you can per subject.
Yeah.
And there we looked, and I was like, well, the thing is, I guess whatever decision we make, we have more freedom here.
Yes.
On the
Yes.
flip side, it comes down to how you're going to use the data, why. And then the Tim Lauman paper came out, which really showed it quite nicely and really solidified
Yes.
in our minds that that we needed more data. And even in the initial NCAT Rockland, there we have 25 minutes of data per subject for resting state, but I couldn't sit there and decide. We were like one of the first two groups to get multi-band from Human Connectome Project. And I was like, what are we going to bank this for? Higher temporal resolution or higher spatial? And so at the time, we wound up doing one scan optimized for higher spatial resolution for 10 minutes, one for higher temporal resolution. Camera got me slowed down to 645 milliseconds, because the more rapid ones, he argued, you're getting eye movements and stuff in there.
Yeah.
And then
Yeah.
Randy Buckner with the Brain Genome Superstruct project, we took their 5-minute scan and put that in there, just so we could sit there and deal with what if the multiband data isn't good, or what if they're concerned.
Yeah.
But
Yeah.
that years later, we'd be like, well, how do you know? We got 25 minutes of data from these people, but there's 3
Yeah.
different sequences, which is a whole other range. So it made the question of scan state seem like the easier one to deal with.
Oh gosh, yeah. You sort of, through all this, you sort of summarized sort of the nature of all of everyone sort of struggles with these things where they see an effect and they're like, oh, is this, and then they're confronted with, like, how do we properly interpret it? And then they have to make a few steps backwards, but also in front of them, they see this like infinite branching of possible ways of analyzing it. And I find, early in my career, I remember just being sort of slowed down, like this inertia of like the possibilities sort of stopped me in my tracks, thinking, okay, what do we do? So yeah.
Yeah, I mean, but that's something. It's interesting. When we built CPAC, the initial idea was, at the time, it was just literally a reflection. I was tired of I didn't think scrubbing was the brightest idea as the primary analysis. I thought there are other ways. You
Yeah.
always knew a reviewer was going to ask. I'd have Claire Kelly would always be on me about, well, we have to do it both ways, and why do you have to do everything both ways? and, and I was like, so. Actually, at the time when I first came to CMI, Satra Ghosh and Arno Klein happened to be visiting. And
Yeah.
Satra at that time was in the early days of NiPipe. And Arno had just published his systematic comparison of differing registration algorithms for structural brain imaging. And so then that kind of inspired me, like, well, why don't we leverage the fact that NiPipe had iterables, and why don't we make it where you can have a very broad number of pipelines? Like you could create hundreds of pipelines. You could iterate over your decisions, which then became its own conversation of, do you really want to have this space where people can choose? So now you're pipeline hacking. And I was like, no, you should know up front which one is the one you prefer. But you could say, how robust
Yeah.
are my findings? And it was
Yeah, which is an important thing. Yeah.
yeah, well, and so some years later, Greg Kerr at the time was doing a master's with Joshua Vogelstein, and so they were the first ones to kind of take what I had in my head a bit, and they took CPAC, and at that point got it to work to create, I think it was like 64 pipelines, and very systematically the different parts of the pipeline, and ask the question of how reliable it is. But Joshua, being Joshua, was like, there is no good reliability metric. We should be thinking multivariate, non-parametric. So
Yep.
Joshua then went through the process of developing discriminability, but it was really one of those moments where we are like, okay, this is actually cool, because you can now figure out which ones seem to drive most or not. And the answer always winds up being global signal, which is like, okay, now we're spending a decade talking about this. Now I feel like we're almost at 2 decades. But the interesting thing was, now years later, so now Greg, he's been at CMI now, I guess 3-4 years. And he's, after Joshua, he worked with over at Montreal with Alan Evans and Tristan, and they basically his work started when I first reconnected with Greg. He was looking at just computational noise essentially, and how you could leverage the variation for purposes of improving biomarker
Yeah.
detection. And that's where he started saying to me, well, why don't you think about ensembling? Like, you have all these pipelines, there is no truth. Why not use ensemble approaches? And that's something that Greg has eventually took over development of CPAC, and really has driven it more with the idea of, okay, there's a multiverse, analytically, but you could actually leverage that variation in a way to try to come up with better estimates of what a ground truth is,
Yeah. Yeah.
and connects those questions. And that's something that he's been working very, very hard towards. And actually, he just put a paper out, also, just really trying to challenge people to accept the heterogeneity and all the variation, and leverage it, which I
Yeah.
think so often people view it as a source and so often it's our enemy. But if you actually say, well, let's take the variation and actually make it what we're after here,
Yeah.
which,
Yes.
if you think about it from the cognitive neuroscience perspective, going way back to like 2008, the Claire Kelly paper back then, we were saying, just look at the coefficient of variation in behavior.
Yes.
Back then, there was so much pushback over it. Reviewers never appreciated the value of I mean, it was very interesting the amount of time Claire's paper got pushed back or rejected inappropriately, until eventually
Really?
it's through a Neuroimage, and people really saw it. And I would sit there and be looking at the reviewers attacking us for caring about the variation behavior. And yet
Yeah.
I'll never forget, then I go to Cog Neuroscience, I think it was that year, and I see Randy Buckner presenting, and Randy was saying, this is one of the things that we should all be thinking about, is variation behavior.
Yes.
And I
Yes.
was like, oh, when the hell is it going to take for a field to get that one? But then we worry about variation. And then when we talk about variation in pipelines, we're very bothered by it. And then now I think we're more into, well, let's learn from the variation. And let's,
Yeah.
especially start digging into massive data sets. It's like, go after, find those points of variation and focus there a lot of your efforts. Don't
Yes. It's
focus
not
on.
just random noise. It's almost never random noise. You're
Mhm.
right. It's correlated with something interesting. And yeah, that's great. So let me just switch gears a little bit, but still keep along the same track, since we're getting into sort of an area that I think is still relevant. I've talked about it in a different podcast, but maybe starting with you weren't on this paper, but this Merrick et al. Paper, and it was sort of a sobering paper, but it was an outstanding paper, and they were talking about sensitivity in terms of the goal of, it seems like, I mean, obviously you have many goals, but and we'll talk about your databases in a little bit, but you have all these databases, and it seems that one of the primary goals is you want to not only derive an understanding of individual populations in different ways, but you also like to have a biomarker that you can then, ideally be able to put it, an individual subject in the clinical scanner and make some sort of prognosis or diagnosis using fMRI about their psychiatric disorder. And what Merrick sort of suggested is like, wait a second, you need 2,000 subjects to even begin to draw any inferences using resting state, or even I think they had a task data, and then a lot of people discussed this, and it really, some people gave up hope. Some people said fMRI wasn't sensitive. I disagreed with both. I think fMRI is fully sensitive enough. And then the question was, is the variation, as we were talking about before, in somehow how the brains were registered and parceled, and is it just all smeared out in that? Or is the variation, as you suggested in your more recent papers and even further back, is it the variation, as you're saying, in individual subjects in how they're diagnosed? I mean, they just sort of lumped everything into psychosis. If you further delineated this, would the contrasts suddenly go up? And then obviously the other dimension is behavior, as we were talking about. That's another source of interesting variation that could reduce sensitivity if you don't monitor it, but it enhances sensitivity if you track it. So, what are your thoughts on this? And then the big question is, are we going to ever achieve biomarkers in this sense?
Yeah, no. So no, I'd say there's a number of questions packed in there.
Yeah.
The first thing I think folks need to do is take a step back and just think of where's the sources of variation that you care about when you're designing a study.
Yeah.
So and from there, people love talking about the imaging sequence and put so much effort on the imaging sequence. There's less effort put on time of day, which is kind of ironic when you see growing appreciation, whether it be global signal or any of the traveling waves or anything that you see emerging in the literature, all highly sensitive to arousal. And, that part there is a little ironic.
Yeah.
But you could sit there and then you could get straight down to how someone's sleep was last night. You can get a whole range of behavioral variables on subjects that we don't control. But that's even just about their behavior in the moment at the scanner and maybe nights before. Then comes the question of what's your actual sampling? Over the years, over the last probably like 7, 8 years, I've worked a lot more closely with Kathleen Amerikangas, who's an epidemiologist. And part of that's been really just, on the one hand, I went up with a bunch of papers of epidemiology and public health, not imaging, which I never thought I'd see in my career. But part of it is really an appreciation of the need to infuse epidemiology in neuroscience, which that Tomas Paus has been saying that since like 2010.
Yeah.
Tomas is one of the ones who was coining population neuroscience, and basically, if you take a population level approach to the game, it makes you suddenly realize, wait a second, who are we actually sampling? And when you're dealing with small samples, the truth is people don't care.
Yeah.
They can't afford to care. And when you deal in clinical samples, sadly, recruitment is hard.
Yeah.
If you care a lot, by the time you get done through all the different realities that you hit, your sample can become more. But I think you first have to start off with who am I sampling, and the most notorious thing in the literature is obviously just simple like age. People talk about autism, and they make inferences, whether it be an adult, an adolescent, a child. Adriana DiMartino, when she first pulled together Autism Brain Imaging Data Exchange and Stuart Mostovsky, you quickly, if you take a look at either of those two Abide papers, you can see the differences in the age ranges used within across studies. And
Yeah.
overly, people often make inferences about autism or so forth that don't necessarily give that perspective.
Yes.
And the same is true for so many other populations, straight down to general population. We make lots of inferences, but we don't think of, so the most basic thing, obviously, is age. When we think of biological sex at birth, that's something I'll never forget. Probably about like maybe 7, 8 years ago, I was invited to speak at what was it? It was the Women's Brain Initiative at fMRI at Brigham, and I was like, they wanted me to talk about sex effects, and I was like, well, that's not usually what I think of so much of myself as a researcher looking at that. But the reality is because of starting with the Biswell paper forward, it's always been there. The reason why we put sex in the Biswell paper is just because if you talk about biomarkers, anything of that sort,
Yeah.
just if you go to lab tests, you always think about sex effects, the different
Yes.
biological sex at birth.
Yeah.
And I was surprised. I expected to see the aging effects in that paper. That part was no great surprise. But when we
Yeah.
looked at this at the sex effect, I was like, oh, I was like, this is. And I remember like a year later, we were sitting there talking to a reporter who was asking us about another study because they saw the Biswell paper, and Claire Kelly and I were sitting there, and she was like, "You're not going to say something foolish about this." I'm like, "I'm trying not to." And the person, whoa, a woman's brain is less connected here. I'm like, and more connected there. What does that mean? I was like,
Yeah.
these
Yeah.
are. I was like, but you have to know the differences. And and then look at Sion Zuo. Sion's done a good amount of work looking at registration errors that come with when you look at an Asian brain versus
Yes.
Caucasian, which in the initial launch of the Thousand Functional Connectomes project, someone basically said there was something wrong with the Beijing data. And Yifeng Zhang said the registration differences you're seeing are probably because of differences in the template.
Yeah.
Not that there's
Yes.
something, but like there's all these pieces that folks don't think about. And
Yeah.
then comes, I think, one of the biggest successes of ABCD is actually drawing attention to the impact of social determinants on the brain and its development, which we had started playing. There's a bunch of stuff we actually never been getting to publish just because one of the folks who led it had left, and there's other things going on. But we spent a lot of time trying to do neurosubtyping in ABCD, looking at brain relations with depression, struggling so much. And then we kept going back to why. And then one day I was like, well, just make sure you think of all the demographic variables, everything, all the prior. And that's where we went coming with social determinant profiles. Those, and those wind up explaining a lot more variance than what people wanted to think at the time.
Yes.
And
Yeah.
but then if you look at medicine, it's always there. I mean, any part of medicine you look at, social determinants will be one of the large effects as far as who's going to get the more severe form or whatever form. So like, it's not surprising, but it was for imaging. And I
Yeah.
think now when you start saying, so wait, what's the value of big data? Well, big data is supposed to let me get heterogeneity from the population perspective. So thinking about the varying social terms, it's supposed to let me understand the variance related to age. It's supposed to let me accept the fact that there are differing etiologies of ADHD, autism, and so forth. And the fact that there are varying etiologies might have different phenomenology associated with them. And
Yeah.
that's all variance in my data. And then comes all of the fun stuff that just varies that we can't control, which Xiaogan Yan, there's a paper called Standardizing 1,000 Functional Connectomes. He had a figure where we listed out every source of variance back in 2013 that we could think of.
Huh.
More recently, Greg Kerr and Ting Zhu have papers on reliability that also revisit that style of thinking. And it just, there's many sources of variance. So
Yes.
big data allows us to do that. And then you have to ask yourself, am I using the right big data set for the question I'm asking? Which, if you look at Human Connectome Project and ABCD, those are meant to be population studies. They're meant to have a level of representativeness, maybe enriched in the case of ABCD. But so then you got to say, am I using the right measures to do that? So Jim Swanson, who's the creator of the Swan many years ago, talked me into the fact that population studies in imaging would go the wrong direction by using measures from psychiatry. And that measures that just say how pathological you are do not work if you have a sample where the average person is without psychopathology.
Yeah.
And because you can't just say, what are my weaknesses? So Jim is a person who in ADHD world created the SWAN, the strengths and weaknesses
Okay.
assessment of ADHD. And, he allows it to be a symmetric distribution. You can be really good at something, not just have an impairment in something. So you have strengths and weaknesses. And when we designed Healthy Brain Network, we actually created an extension of SWAN called the eSWAN. This was Jim, Giovanni Saloom, myself, and Lindsay Alexander, where we took several differing disorders and showed you could take the symptoms from DSM and make it where you have a trait dimension that goes from strength to weakness. If you have that, now you have meaningful variance across the full population. That's different than what the population studies will go to when they take CBCL or adult self-report. Because then what's happening is you wind up with a measure that for much of your sample has zero variance, which
Yeah.
if you have that for zero variance, now what you have is a truncated distribution.
Yeah.
And we actually showed this in the ESWAN paper just in the behavior front when we were validating those scales.
Yeah.
And this is something that when people look at that Maverick paper, I have so many people who are like, see, so you could pick up cognition and intelligence, but you can't pick up psychopathology in the brain because this and behavior. It's not. And I'm like, wait a second. It's actually not a reliable measure. And the thing is, they try to point out the reliability of the CBCL and Occubox measures, and they say, see, it's like 0.91. That's actually not accurate. If you go back and look at the paper they're citing, in that paper, what you'll find, Aki Nicolaitis and I had done this some time ago,
Yeah.
is the reliability is not based on the thousands of subjects in the study. It's based on a small subsample
Huh.
of individuals who, if I remember right, had average or higher levels of symptomatology.
Okay. Okay.
And this is something we've published on this kind of stuff in actually in Toby et al. It was an NKI Rockland longitudinal paper where if you look at the correlation structure of the CBCL in your full sample versus put it towards the clinical,
Yeah.
you're going to see a notable difference. But you should, because the full sample has lots of zero variance.
Yeah.
And so you have a truncated distribution.
Yep.
And then you're going to say, look, it does worse relative to cognition, which we've seen that in our hands too. It's true. You
Yeah.
will. It's a population study. You probably should not have been asking the question using that tool there, which, that's not a criticism. I mean, it's something that is part of the learning experience that we're all going through.
Yeah.
And, it's when we're designing population studies, we need to think a lot more about the phenotyping and what it is that we actually want to measure.
Yep.
And I mean, we're all guilty of that in any of our studies.
Yeah.
So it's something that, but we have to be careful because then you wind up with some inferences get floating around, and you're like, no. It does not mean behavior does not map to the brain.
Yes.
It's pointing to the tools that are being used to characterize behavior in my mind. And
Yes.
then comes the question of, even if you did have a good symmetric distribution, does that solve it?
Yep.
Probably not, because you're getting now into the challenges of mental health phenotyping and behavioral phenotyping. And that's where, when you look at the literature, we know on the one hand, these tools are designed so that mental health practitioners are reliable with one another. And they're designed to be able to help us to monitor changes at a broad scale in an individual. It doesn't mean that that's what's going to let us get those great insights into the biology.
Yeah.
That gets into the whole issue of straight back to DSM diagnostic labels.
Exactly.
Yep.
Yeah.
And the questionnaires we use to assess.
Yep.
So I mean, that's where, if you look at, if you look at it from that perspective, you have folks like Kathleen Merkangas, who really has been the lead in moving the field towards ecologic momentary assessment, where it's really about, let's look at the variations in someone's behavior over the course of a day and across days. Because a lot of the information about mood disorders, for example, is present there, and get that in a questionnaire.
That might be interesting.
So that's really been getting us to think differently. So we spend a lot more time now trying to develop methods for doing real world, more ecologic style assessment of individuals, whether it be symptoms, cognition, so forth, and trying to think through how do you pull that in, as well as how do you also do more multimodal characterizations. So that goes heavily influenced by Scott McKeague. Scott, years ago, Healthy Brain Network, he reached out to me and said, "You realize you have EEG and eye tracking simultaneously in a lot of your subjects?" I was like, "Yes." Simon Kelly and Nicholas Langer had worked with us, and Lucas Parra, and helped develop those protocols. And he's like, "That's really valuable, but you have it in a large sample. But what if you had more modalities?" And he started pointing out mobile brain body imaging and his whole literature about naturalistic cognition, which again goes back to 2009 or 10. And Scott was really just in his usual fashion way ahead of everyone's thinking, and basically saying, how are you characterizing cognition? You could fully characterize it in its entirety
Yeah.
and really got us excited about really stepping more into that model of simultaneous data acquisition, which we've really not, like the new NKI Rockland sample 2, which Stan Colco and myself are leading there. Yes, we have multimodal imaging and we put some of the latest and greatest updates and so forth, but we actually have people doing Mobi sessions where they are sitting there with eye tracking, with EEG, EMG, EDA, motion capture, respiratory, cardiac, all while sitting there on a tablet with a platform we developed called MindLogger. They're tracing spirals. They're
Yeah.
sitting there writing. They're doing different tasks. Also, that you could get everything simultaneously, get these high def characterizations of folks, which then
Yes.
vision is some of those same tasks we get folks go do on a mobile app at home or on the web,
Yeah.
building towards that vision of trying to connect the pieces. And then
Yes.
it comes the bear of taking on fMRI EEG with the many different sensors and so forth, which we've been suffering through at NKI. And
Yeah. Yeah.
but I think there's so much opportunity that then when you look back at the Merrick paper, I mean, the idea you need large sample sizes is not that one's not new to me. My thing is 10,000 actually feels small if you actually take into account all your sources of sampling variation, which is not
Yeah.
something we should be apologetic about. It's
Right.
what reality is. It's what drives us. I think whether it be increased focus on sampling, whether it be increased focus on characterization of behavior cognition, and also then comes the other part, which is increasing really making sure we're using the right amount of data for functional MRI studies,
Yes.
which that's something that we've known for quite some time. You need to really, get up to 25 minutes, unless,
Yeah.
I know. So there's the work by Connor Liston and now more recently Damien, and obviously we're doing a lot with yourself,
Yeah.
multi-echo and multi-band multi-echo, and
Right.
really are making these arguments that you could do so much better with so much less, which would be great to, if that's true. And we, like NKI Rockland's data, we have multi-band multi-echo there,
Yeah.
and we collect, actually you helped us with some.
Right. Exactly. Yeah.
But
Spreading the
so
word.
it's,
Yeah.
I think there's reasons to be excited about where the field's going, but I also think it points out a couple also confusions that we've all had over the years about what are the low-hanging fruits for imaging.
Yes.
Low-hanging fruits for imaging, I think Mike Fox is one of the ones driving home the low-hanging fruit. Like,
I
if you're
totally
doing
agree.
a intervention, you should
Yes.
really study the brain and measure the brain. I think Mike has been brilliant with what he's been doing
Yep.
in the brain stimulation literature that way. I think that is predicting diagnosis. It has potential real world relevance. I don't know if that's really the best deliverable right now, as opposed to just what's more accessible in our minds, because the best deliverable would be predicting outcomes. When you
Yeah.
think about precision medicine and when bringing that to mental health, that's really about, let me figure out where this is going and which intervention might impact things in the most meaningful way. That's not
Yes.
necessarily about diagnosis. It's about, if I have all my varying measures, then I can make that. But the challenge there is you need longitudinal data.
Yeah. Yeah.
And so, but I think we make it a little more intractable in certain ways by saying, well, we need longitudinal imaging data. We don't necessarily need that. I think longitudinal imaging data, its value may be in understanding the longitudinal data may itself be more informative from the brain by seeing trajectory, and that could be your predictor. Then you need longitudinal
Yeah.
data.
But you only need one time point, really. I mean,
Yeah,
yeah.
but
And
what you needed is good follow-up data. So now you need electronic. You basically need either apps or something that allows you to stay in touch long periods of time, or you
yes.
need to be able to integrate with electronic health records or other ways of capturing data.
Yeah.
And but with respect to outcome. But
Yeah.
now what happens is you need to get real world, people who are really dealing with medical research and thinking about the outcomes that matter most in the field, and marrying them with folks who are approaching this from the neuroscience and so forth, which that's where there's a lot more need for interdisciplinary connectivity and
Yeah.
and also just the technology. I would say the biggest game changer of the last 10 years has really been the biggest game changer of the last 20 years, which is the phones. You know,
Yeah. Yes.
going back to my story of how I did in residency and fellowship, if Steve Jobs didn't exist, I don't think my residency or fellowship would have been close to successful. I just had like a Palm Pilot that wasn't very good, but
Yeah. No,
once I
I've
got
had
the
Palm
iPhone,
Pilots.
yeah, once iPhone, I could sit there and be on top of everything and very
Yes.
quickly. But
Yeah.
now, what's happening is that we could get data, that we could start getting voice samples. You know,
Yep.
when Arnold Klein first came to CMI, Arnold was at the time working at Sage Bionetworks, where even though he was an amateur, he was helping in an effort to get 10,000 samples of Parkinson's individuals with biometrics via phone.
Yeah. Yeah.
And that really resonated. And I
Yeah.
think that the phones are, and especially now in the last 2 or 3 years, you can see what's happening with LLMs emerging.
Yep.
We
Yep.
are seeing an acceleration in what can be done, also
Yeah.
all the different perils that come with that.
Yes.
But I do think we're going to get better with the characterizations. We're going to get better with the longitudinal data and with those, and also building up incentives for people to participate and lowering with the barriers.
Yeah. Let me ask so um, there's a few places where I had to I have to add something or maybe suggest a question. So you were talking initially about, right. I totally think that the future of psychiatry, it seems like it's stalled, but basically if you just have a doctor sort of making an assessment based on the DSM sort of assessments, everyone's trying to map the DSM to the brain activation or the brain patterns, which may or may not happen, and it probably, and it seems to me that the future, as you said, is this multimodal sort of real-time measures over time with a phone, with some sort of metrics that then would give a much more, nuanced picture that could be much more accurate of pathology of some
Mhm.
sort. And then then the hope is that you could use the neuroimaging data, which seems like it sort of catalyzed this because we're not getting satisfying results yet. But if we start to further delineate and further take some sort of a multivariate approach, there might be more likely success in characterizations. It might be not require quite as much data, but still the more the better. And yeah, so I think that is that correct to say that right, one, the future I think is in this multimodal, but two, that if you're trying to come up with biomarkers, it's like you addressed you talked about all the issues in acquisition and processing and normalization, but a lot of it could be, like you said, that we're not really capturing or delineating the differences in the pathologies in some sense, and so it's sort of averaging
Yeah,
out.
no, absolutely. I remember years ago, Joshua listening when we were talking about this one night. His thing was, so what you're saying is your one side of the equation is wrong. It's no longer the imaging. I was like, well, it's an oculonicolitis. It's still only a preprint, but he has a paper that really delved into this very much, thinking about things like joint reliability and so forth. Both
Yeah.
your measures need to be reliable. Both measures need to have the level of validity needed for the purpose you're chasing, whether it be clinical or scientific. You need to think about both sides of the equation. I do think the big data, historically, we all thought about big data as far as sample size because we were so plagued on that front. But now I think we're understanding really the truer value of big data, which is really about capturing the heterogeneity and
Yep.
also the need to just keep increasing the quality of data. If you think about it, all of the open data, pretty much, with exception of the NIH funded large scale studies, almost none of it has 25 minutes of data. Anything you see from
Wow.
ADHD 200, 1,000 functional kinetics per none of those, which talk about thousands of papers.
Yeah.
And they have
Yeah.
fared well despite not having the ideal amounts of data, but they did that because of the larger sample
Yeah.
sizes. But this is
Yeah.
where, Senyan Zhu had a paper, harnessing reliability for neuroscience research, where this was back in 2019 in Nature and Behavior, where Ting showed some beautiful visualizations showing very clearly you're making a trade-off. If you increase the reliability of your data, you need less number, you need fewer subjects. And
Yeah.
and I know Tom Nichols was a part of a collaboration. I forget who the first author was, which really, I think, more recently showed one of the more elegant presentations of sample size calculations and thinking it through for imaging. That was
Yeah.
presented this year at OHBM. I think the field is getting there and figuring
Yeah.
this out and appreciating it. But then it's a matter of putting it into effect and also dealing with the fatigue that people feel.
Yeah. Yeah.
The thought that, okay, now we need a new wave of studies. Everyone's like, oh gosh, are you kidding me? But the thing is, we can still learn from what we did. I always get driven crazy when people say, stop research until you can figure this out. It's like,
Yeah.
no,
No,
that's not how this is going to go.
that's not how
But
it
at the
goes.
same time, like when we know something, then we've made those jumps. We do need to now update the models. We need to, and we need to be okay with that. And
Yeah.
it does, research, I think, these more intensive designs. And that was honestly, one of my biggest take homes at OHBM this year was that whether you're looking at pain, mood, or anything else, it seems like we increasingly have the ability to make individual specific predictive models.
Yes.
That
Yes.
really seemed to be a message.
Yeah.
Group level, that not so much.
Yep. Yep.
But that is maybe okay. That's an okay starting point. If we can really figure out the individuals, then we need to understand what are those sources of between subject variants that are so
Yes.
problematic. And from that perspective, that lives at every level we discussed. But if
Yeah.
we really do better in understanding every level
Yeah.
between subject variants, we'll eventually get there with that. Plus, if you start stacking individual level models, because even when I think about with the phone apps, I think people are trying to do diagnosis and all this kind of stuff. I'm like, oh God, don't do that.
Yeah.
Take the
Yeah.
value. You can look at someone's variance relative to themselves,
Yep.
know something about that person. So if you see a person with bipolar is speaking faster, that matters more than a person with no history of anything. And so
Yes.
you have some knowledge, but look at, are they speaking faster relative to themselves? If so, you can make actionable decisions. And then because of the scale of the digital technologies, you can stack those. So you
Yeah.
eventually do get those large scale data sets. And the same,
Yes.
I think imaging, if we take that step back now, and we really figure out how to do it really well for at individual level, leveraging everything we've learned from the big samples and all the techniques and everything. And then we figure out how to get to that next level, which is the more
Yeah.
generalizable models, which that's just a matter of time once we've really worked out the first piece.
Yeah. Do you think though that then I immediately jumped to this deep sampling. I mean, do you think 25 minutes is enough? I mean, what about 5 hours?
That's a fair question. I think I think if I had Adriana DiMartino here, who is an autism researcher and is the one that sits there and works hands-on with the kids, and I said 5 hours, I think you would get a violent reaction.
Well,
I think it's. I think there's a couple answers. One is if multi-echo really can bring down those reliability requirements and multi-band multi-echo and putting in Nordic and everything else at hand
yep.
does that, we should absolutely figure out how to do that because there are going to be
Yes.
populations, the more impaired populations who probably need imaging to be more useful than others. And they're going to, we're going to need to make it more tractable that way. And some part, of course, is analytics. If you look at Vince Calhoun's put a lot of time, Christian Beckman, like so many people are trying to figure out how do you do the most with the least data
Yeah.
and keep getting that way. With that said, I think you got to, going back to like our experience with the Healthy Brain Network, the serial scanning initiative, if we didn't sit there and literally wind up collecting, what was that per subject, it would be, I guess 12, yeah, it was 12 hours of data, 1 hour per condition. If we didn't have that amount of data, it wouldn't have given us the insights that we had. And we've played around with all sorts of analyses trying to figure out with what amount of data does pipeline variation or different decisions not matter?
Yeah.
And those style questions.
Yep.
I mean, once you create the data sets, you can answer those questions. But also some of the questions are humbling. Like with Ting Zhu, when we first stepped into non-human primate imaging, Ting had a paper that we did with in collaboration with Damien Fair's group, where what we did is we were getting non-human primates, and one of the things that's interesting is when you look at reliability, you can be very reliable to, when you looked at it was like, if you looked at them while we were doing, depending on their variances in awake state, that the variances in wake state, you can still see very similar to one another. And then comes awake versus anesthetized. And you can be very reliable awake, very reliable anesthetized, but they're different states.
Yeah.
And so you now have to take that into account when you want to make inferences across states.
Yes.
And, and then when we think about medications in humans, we don't really have a lot of work looking at really what's the impact of giving someone a neuroleptic on the image. It's always seen there as a confound. That's a real world challenge because it's really hard to give neuroleptics to people who don't need them for all the
Yes.
obvious reasons. But like these style questions, I mean, there's just so many pieces we would have to fully address. But I think if we create these really deep, dense sample data sets, we can really work to just figure out what are the minimum requirements. I mean, for us, we're always thinking about what's the minimum necessary data, what's the minimum necessary data, and I think that's the right mindset. It's just we have to be careful because the ambition can change.
Yes.
On the
Yes.
flip side, one other caveat I would give is back on the big data side of life, where like these large samples, like so Ted Sathwaite and I recently released the Reproducible Brain Chart project, where that is 6 sites, data that we went through. Literally, we thought in 6 months we have the first version out. It's been over 4 years. But we literally went through having to deal with all the questions, pipeline harmonization, data organization. Ted created qubits and varying structures just to deal with some of what we were finding that you would never expect, like off a single scanner, seeing variation in metadata and DICOMs for no
Okay.
explainable reason.
Yeah.
And then once we dealt with all that, we also had to get Giovanni Salum and his team and to sit there, and we used a bifactor model there and said, okay, we're going to go into a bifactor space. We escape all these things, but literally 3 papers to figure out how to deal with questions of measurement and variance. So does the CBCL at each of these sites mean the same thing? Because one's in Brazil, one's in China, one's multiple in the US. And then
Yes.
what you do when one site doesn't have that measure, it has others,
Yeah.
and all the problems, like these are parallel paths. So I think we need to be thinking about the really optimizing the daylights of what we could do with the imaging and really getting those very strong individual level predictive models, while at the same time, and figuring out how to generalize as best we can. But part of that answer is going to be dealing with all these questions that we can answer with these large scale data sets
Yep. Yep.
about harmonization.
Yeah.
Adriana DiMartino right now has her autism mega analysis project where she's working on taking not only all the open data that through the Indian Autism Brain Data Exchange, but she's also going through ENDAR and everything and dealing with all the harmonization challenges.
Yes.
That is hard. But
Yeah.
if we want to think of how not to be in the same challenges 5-10 years from now, we literally need to work to deal with all the phenotypic challenges and get people to actually agree to acquire data
Yeah.
in a way that makes it easier to come together, which that should be easy, but it's not. I know even when I have collaborators that work very closely with party A and B, and I try to say, can we just, it's often the answer is it takes a lot of work.
Yeah.
Pull folks together, not because they don't want to work together, because they have different research questions and it's
Yeah.
going to, create different biases in how to do it. And you can't say
Yes.
right or wrong.
Yeah. No, actually, you're right. You're right. And also it might also be possible that even with deep sampling and you're collecting more data for an individual, right? There's different questions with the big data questions, and then you have to definitely solve those if you start looking at individual sample data that's sensitive enough to pull into that. But what about, I mean, I often think that the more you collect the data and the more reliable it gets, there's ever more nuanced results that you might find. It just seems like a continuum. I mean, it seems like we haven't found some sort of we haven't. I don't even have a sense of any sort of limit in of.
That's a great. So there's a paper that we have that came out last year, Leonel, where we did this was a collaboration looking at pipeline differences. And this started actually as part of Reproducible Brain Chart project, where I basically looked and I said, well, Ted and I were looking at which pipeline we use. And I was like, well, CPAC's configurable. So why don't I just make it configurable to be able to overlap with the fMRI prep and XCP, and that'll be easy. So a few years later, we're and we were looking, and then you have Damien with the ABCD pipeline, and we spoke to Chao Gan, who of course has DPABI and DPARs. And so we first played that game of just running the same exact data set. It's openly shared, the HNU data set from the Consortium for Reliability and Reproducibility project, where same data set through every one of these pipelines. And I remember at the time I was like sitting there saying, what might be a good number? And Russ is like 0.9, maybe 0.8 on the board. We're like looking, and I'm like, oh. And it's very humbling, because then we had to sit there and figure out why. Like, why are we seeing fine some connections here and that, but a lot of it is going down low, and some combinations of pipelines
Interesting.
really get lower and lower for some connections.
Yeah.
And one of the things that was interesting, so at first, like, well, this really does not feel good.
Yeah.
And the fact, this was all the minimal pre-processing pipelines. This is where I'm doing exotic stuff. This is literally, we took 5 different pipelines. They're minimal pre-processing. Went through all this stuff with comparisons. This was almost, I felt like my lockdown project, because we were like sitting there, and I'm like jumping online, being like, show me this now, show me this now. And we were all sitting there going through this. But then came the question of, well, how does the amount of data that you use, because the HNU, they had 10 minutes of data across 10 sessions. So you could get up to 100 minutes of data.
Yeah.
And so it's like, how does that improve anything? And the answer is, it improves what you expect it to improve, but not the problem you're looking at. So it improves the reliability of it deals with the test-retest reliability question. But what it does is, it gives you both the sobering message and maybe the hopeful one for the literature. The sobering message is that as you increase, all the effects that you see of pipeline when you're taking same subject, same data, if it's same subjects but different data sessions being fed to different pipelines, they mostly go away, because the data has with only 10 minutes of data, your reliability is more impacted by the test-retest than your pipeline.
Yeah.
Go and you crank up the amount of data that you have per subject. Now you can appreciate these pipeline-related differences.
Yes.
So then I looked at it, I was like, well, so the good news is we're not in trouble for yesterday's data, because yesterday's data hasn't hit the ceiling. But what you're seeing is the pipeline-related variation. That's the new ceiling on reliability when looking between studies and different analysts and so forth.
Yes.
So that was the uplifting part of the message. And then of course, we went through in CPAC, and we played, actually I modeled it after, this is going to sound silly, but antidepressants. We have, for psychiatrists, we have these charts of switching from and to, and the decision making that goes for each.
Yes.
And it's like, well, why don't we come up with a chart of from and to for pipelines, where and then within there you're looking at, if I switch from this decision in this pipeline to this one here, and you can do that individually, and where does the error, where does those variants arise? Which the answer is comes up being depends on which pair of pipelines you're looking at.
Oh God,
but, it was a fascinating endeavor.
But pipelines aren't necessarily. I mean, hopefully, eventually, we'll understand the reason for the pipeline differences, and so get the sort of underlying principles, so we can sort of, reduce the variability in that sense, instead of just, having this pipeline diversity.
Like for instance, I'm saying there's a question of to what degree do you ensemble, to what degree do you look for what's the common, and also to what degree is it that you always make sure you at least have benchmark. So there's the EndMind effort that this Greg Kier, Damien Fair, and a whole bunch of us put together a few years ago, where it was this idea of trying to get the teams communicating, but also can we sit there and even come to agreement among groups that are friendly with one another, how to refer to different steps and how to think about our documentation, evaluating examples of code. And there are these hackathons that are happening. There's a piece that was put in Nature Human Behavior, it's kind of call to action for this. And really thinking through, the questions of testing at the differing levels, and when we're, as when it comes to references, is there some set of data that could be used as a reference data set? And you could say, we at least tested these pipelines. We
Yes.
know behavior on this set of data sets, how it differs from one another. Here's
Yep.
what we don't know, and those style questions. And we put a lot of thought into that as far as where the field can go.
Yes.
At the end of the day, if you can at least understand the connectivity between the packages and figure out what matters, what doesn't, and to your point, why.
Yeah.
Sometimes you can address the why, and sometimes the why winds up being data quality. When the Nature and Behavior paper was talking about with the pipelines, there we said maybe it's because we're using the HNU data and it's just old data. But then we used the Healthy Brain Network data, which is based on ABCD, and we're like, no, same issue, still there. But it also it's interesting. You can it's those kind of investigations are not very appealing to do. They take an incredible amount of time. You go down a lot of rabbit holes, but they can be informative. I think there's at least a part of the field that needs to dedicate itself to that.
Yes. Yes. It's one of those things that researchers don't even think about when they're, I totally agree with that. And it would probably help a lot in and like you said, having, who knows, some sort of grant mechanism that says, oh, this is a category of things that sort of lay the groundwork in terms of pushing the field forward. And those are overlooked a lot. Yeah. I mean, it reminds me a little bit of what you're talking about is related to the Botnovic Nezer paper, where you had one data set and I guess 70 or so people analyzed it.
Yep.
And they all got different results. I mean, and then obviously there's an argument that if you lower the threshold that they kind of converge, but the message was clear that there's enough variables that people use that are unaccounted for that pipelines use and in the order of variables. And like you were saying, it's still somewhat uncharted. Yeah.
I mean, look on the bright side, the imaging community, for all the different things that have been said about it, we've gone through a lot of mature to a place that there are many fields of neuroscience that are just realizing they're going to need to go through the same process.
Yeah.
And hopefully the same way Washington genetics community has been an inspiration for us, hopefully our community's growth will be inspiration for others.
Yeah.
But that process is unavoidable, and we're not done with it, which is okay.
Yeah, and that's a good thing. That's it's exciting. So, right, and I don't know if I want to. Well, I'll briefly bring it up, but first, before I do that, you mentioned a lot of your databases. Instead of going through all of them, I mean, you're associated with so many, and it's just amazing. And the most recently, I guess, is the 10,000 participants CMI Healthy Brain Network that finally has converted.
Yeah, there's the Healthy Brain Network, and then probably the most recent is with Tess Hathaway's the RBC, which takes like NKI Rockland, Healthy Brain Network, Philadelphia Neurodevelopmental Cohort, the Brazilian High Risk Cohort, as well as the longitudinal sample was developed in New York by Nim Tottenham's team, and the Chinese CONEST project's data, and deals with harmonizing it all in order to then make it available and downloadable. And that there we're just tying down. The data is open. It's just that we're tying down the initial manuscript to put up on bioarchive, to which I think will make it a lot easier for folks to really understand what's there or not.
Okay.
But these resources, I think with each one of them, what we're trying to do is create data to help folks move forward.
Yes.
When one looks like NKI Rockland or Healthy Brain Network, those are examples of ones where we saw there's not data that exists to address the challenges. So there we need to create these data resources to really let us ask the questions that we want to ask, as opposed to some things like ADHD 200 or Abide or the Primate Data Exchange or Core, where, okay, data is out there that exists. Let's pull it together and let the field move forward with that, while some of these other data sets are being built and really hoping to change the nature of the questions that we can answer.
Yeah. One other thing too, and this sort of, I mean, the goal is, coming up with better ways of looking at, outcomes and basing it on neuroimaging data. And the goal, the outcomes is of some sort of treatment, either drugs or therapy or something. And still in that realm, psychiatry is somewhat, I wouldn't say stalled, but it just reminded me of Tom Insel, who used to be the director of NIMH. He sort of left. He's actually gone the route of massive holding a phone, sort of sampling in terms of real life sampling. And he also he wrote this book basically saying, sort of a confession, like, well, I really fostered imaging and molecular biology to sort of try to intervene and change the circuits or understand the circuits, and which has promise. I still think, like you were saying, Michael Fox's work is promising. But the whole point of that book was like, oh, we should have all our money spent on better therapy, like better. There's something missing there in terms of access to therapy, and where you don't need really imaging, you just need, yeah. I'm not sure. I think there's room for both, and
Yeah, I was gonna say it's more of an and rather an
right,
or. I mean, the challenge is, right now we've been working on developing some digital solutions, whether it be for data capture, for intervention, and so forth. And there, that is its own learning space. Which Thomas, he has a really nice paper he wrote, I guess, a few years ago or a couple years ago, where he talks about Act One of the digital therapeutics, and now then comes Act Two, and how many is it going to take? That's his own land of discovery, because it's not just about digitizing. Like, you can't just, I mean, that was what the field essentially did, is let's digitize the PHQ-9, let's digitize this, digitize that. Great, you got to get somebody to actually use it. So what are you doing to move engagement? What are you changing? If you want to talk about accessibility, the challenge is you can take everything we currently know in mental health that works, put all the effort into innovating and scaling that, and you will move the dial somewhat, but you're going to hit the ceiling because you needed innovation in the way we think about things. You need to innovate technologies. I always sit there and, any of the folk work that we do with the therapists, I always say, think of your best therapist. You either want to know, how do you scale what that therapist does? How do you let that therapist do something they never could do before? How do you just make it more efficient for them in their day-to-day operations? And I worry that number two is not easy.
yeah,
Finding ways to move the needle for them so they can do what they couldn't do otherwise, that takes work. You're talking about really taking a step forward, and you start thinking about closed loop, where you could be interacting with patients in a way that you never thought of, having connectivity with patients, with the
yes,
technologies, and all the leveraging machine learning, AI, all these things. It's not trivial. It's a bit of wild, wild west, which is what we'd say about imaging 25 years ago, or maybe even 20 years ago. This space, it's just at its beginning. There are so many things, but it's more reckless in certain ways because it's already out there.
yeah, exactly, yeah,
Except of a few folks in the field who, for whatever reasons, they did what they did. Most people are not using imaging to try to change clinical practice.
right,
Right now, it's out there, and it's its own set of challenges, but it's also the real world research or ecologic research. Like, you do need to be out there in some way, but you have to do it responsibly. But engagement is actually the number one thing that you
yeah,
watch why all that world flops. So we haven't really figured it out yet.
yeah,
And then comes to make things accessible. You need to really think about diversity. You have tools that could deal with, the diversity, whether within country, across countries, all these, so now you
yeah,
start to think about tools that have not only left to right languages, but right to left languages.
huh,
So many different pieces that you have to think of in this. So
yes,
it's another grand challenge, but they are far from exclusionary. And he knows that. I think
yeah,
that he's trying to emphasize and draw people's attention towards impact, which a lot of times the academic community gets caught up in the varying intellectually challenging debates and innovate, but doesn't always fully think about what's the impact of this is.
yeah,
And in part, we need people to be doing that. I don't
yes,
think it's necessarily a bad thing. I think
no, yeah,
people don't appreciate that. Some folks need to be worrying about all the real world impact, and some people need to be understanding things, why they work the way they do, because once we figure this out, these folks will now see the value of it.
yeah,
But
yes,
you can't be upset if you're on this side, about what you don't know if you were at the same time saying, don't let people go figure out.
Yeah, exactly.
Yeah.
Yeah, that's a really good answer. I like that. It's they both sides need each other for anything to progress. But so anyway, so we've gone much longer than I had even suggested. So I think that this has been overall, really super stimulating, and yeah, I really appreciate all the thoughts, and I think that for people listening, I think it's important to listen carefully because there's a lot of points brought in that are really important, and yeah, I think. Yeah, I'm hopeful about imaging as far as that's concerned, and I'm hopeful about clinical applications of fMRI and also potentially towards psychiatry. And definitely, you're leading the way in this. So I
Oh, thanks.
appreciate
Yeah, no, I think there's a lot to be hopeful for. I think we just need to put things in perspective. And from imaging perspective, we have a broader range of tools to work with, not just imaging. So if we take that view, we take some of the load off imaging for some of the expectations that are not so short term. And we can really work on the long-term value and also lean in on those fronts where right now we can see some impact.
Yes. Yeah. Yeah. All right. Well. Thanks again, Michael, and I wish you the best, and look forward to seeing your papers come out in the future and seeing you at meetings. All right.
Thanks, Peter. Much appreciated.
Neurosalience is brought to you by Organization for Human Brain Mapping. This episode is produced by Ömer Faruk Gülban and Alfie Wern.